Welcome back to this next video in which we are discussing influenza. Okay, so in the previous video we discussed the basis of long-lasting immunity to influenza. So, when you've been infected by a certain influenza viral strain, after that you will be immune to that strain and also a bunch of similar strains to the one that infected you. So, initially the basis for this long-lasting immunity is that within the first few months you're going to have high levels of IgA in the mucus that lines the respiratory epithelium of the respiratory tract, and this will be able to neutralize the entry receptor of the strain of the influenza virus that you were infected with, and also similar strains to it that have similar entry receptor molecules, similar epitopes on their entry receptor molecules. Now, of course, that doesn't last forever. The long-lived plasma cells do not live forever, unfortunately. So this only will be there for a few months, but it's fantastic protection. It's complete protection there for a few months for the strain that did infect you and strains that are similar to it. Now, after that, the basis of long-lasting immunity is the secondary immune response. So the fact that now we have loads of memory B cells, memory CD4 positive T cells, and memory CD8 positive T cells, which means that if we were to initiate an adaptive immune response again to that influenza virus strain, it would be much, much quicker, and therefore the virus wouldn't get as far down the respiratory tract. And this means that you will just get the early respiratory tract symptoms, so the nasal symptoms and the nasopharynx symptoms, and maybe just a few mild systemic symptoms because you've got a much smaller patch of inflammation and therefore much less TNF-alpha, interleukin-1 and interleukin-6 going into the bloodstream. And again, the secondary immune response will not just be helping you out against the specific strain that you were infected with, but also very similar strains to the one that you were infected with, because those will have similar antigenic epitopes and also similar peptide fragments in their antigens, and therefore we'll be able to use our memory B cells and memory T cells against these similar strains as well as against the actual strain that we were infected with. So that's the basis of long-lasting immunity, and it's very important to understand that. So, we're now going to discuss two complicated concepts, antigenic drift and antigenic shift. And I want to discuss now why it is that influenza A virus strains can cause pandemics, whilst influenza B virus strains and influenza C virus strains don't generally cause pandemics. And remember, a pandemic is when it infects the entire world, pretty much, or it infects a good chunk of the world. It spreads just from human to human to human to human and goes continuously over the world. Okay, so antigenic drift and shift is what we want to discuss. So, these are both ways in which you can create new influenza virus strains of all three different influenza virus species over time. So these are ways that we can create new influenza virus strains of the three influenza virus species. So let's start off with antigenic drift, then we'll come on to antigenic shift, and then we'll discuss why it is that in the influenza A virus population you can get strains that will cause pandemics, whereas you don't get that in the influenza B virus and influenza C virus population. So, firstly then, antigenic drift. So, antigenic drift is the concept that gradually the antigens of the influenza virus are going to change. They're going to drift away from one another. And so how does this work? This works because of the fact that the RNA-dependent RNA polymerase of the influenza virus particles is not particularly good at copying RNA. So remember, when we copy our viral genome, the way we do it is through the RNA-dependent RNA polymerase. However, that makes mistakes far more often than, for instance, the DNA-dependent DNA polymerase of humans would make mistakes. So I'll just put this here. The RNA-dependent RNA polymerase is really important. This is the thing that gives rise to antigenic drift. So the RNA-dependent RNA polymerase. So it will make mistakes, and this means that when it is producing new viral genomes, those new viral genomes are not actually going to be identical to the original viral genome that was the genome of the virus that initially infected the host cell. So, when a virus, influenza virus, goes into a host cell and produces thousands of new virion particles, they're not all going to be genetically identical to the initial one. There are going to be small mistakes, and on average, there is one nucleotide mistake per new virion that is created. So I'm just going to draw a little picture of this. So we'll have in white here our infected cells. So here are its cilia, here's its nucleus, and then I will draw on here. Here is the one virus coming in, so it comes in, and then of course thousands of virus particles come out, and that's the basic replication cycle summary. And the cell, of course, dies after it's produced thousands of these new virus particles. So each one of these will have a genome, and the genome will not actually be completely identical to this because this thing makes mistakes. And as I've just said, on average, if you look at each one of these and you ask how many mistakes are there in its genome compared to the genome of the original one, there will be one nucleotide mistake in the entire genome compared to the original one, i.e. one nucleotide in the genome will be wrong compared to the original one. So, this is what gives rise to antigenic drift, because it means that gradually, as you go through more and more replication cycles, so of course all of these will go off and then infect other host cells, and they'll make new virus particles, and it continues on and on and on. So you get virus particle after virus particle after virus particle, but each time that you make new virus particles, there's going to be one change in the genome. Now, most of these nucleotide changes will cause absolutely nothing. They won't change the proteins of the virus at all. Some of them will change the proteins of the virus and make the virus totally useless now. Some of them will then change the proteins of the virus and not make the proteins useless, but will now make them so that certain parts of the immune system can't actually recognize it anymore, change potentially the epitopes of the antigen. And that's the concept of antigenic drift. So because the viruses, when they go through the replication cycle, are gradually diverging from one another, as you can imagine, as this goes on and on and on and on and on, the virus particle in many generations down the line from this one might be quite different from this original virus particle. So antigenic drift is the concept that virus particles are gradually going to change because of the fact that the RNA-dependent RNA polymerase does introduce mistakes, it does introduce mutations into the genome. And this is how we're going to create new viral strains through antigenic drift. Of course, this is a really slow process, so that's a key point. Drift is slow, and it's very, very gradual. So you have an evolution, if you like, a very slow evolution, and evolution just means change. So you have a very slow change of the virus. In fact, I'll draw this out. So here's the first virus, then let's say here's one of its progeny. So this virus infects a host cell, and this is one of its progeny that will have now one little change potentially from this one, and then it goes on. This one will go and infect a host cell and produce progeny. Here's one of its progeny, and it will have another change from this one, and therefore it will have two changes effectively from this one. And of course, it's on average there's one change per progeny, but in reality you might get some new viruses that have more than one change from the original one, more than one nucleotide that is different in the genome from the original one. And it goes on, and gradually, as it continues on, as you get further and further away from the original one, you're going to have more and more changes. So gradually the viral particles are changing, and when they are different enough from the original one, that's when we might start to call them a different strain of the influenza virus species compared to the original one. So this is the concept of antigenic drift, that gradually, as you go around more and more cycles of the replication cycle, and get further and further and further away from your original influenza virus particle, the antigens of that influenza particle are going to gradually change, This is why we call it drift, because it's very slow antigenic change in the virus particles, and eventually we might call this a new strain of influenza virus particles. So we might call this one and this one different strains, and in reality it will take far more than just 4 replication cycles in order for them to be different strains, but I hope this picture might get that concept across simply. So, gradual change is introduced in the influenza viral particles by the fact that the RNA-dependent RNA polymerase does not copy the genomes perfectly, and once enough of those changes have built up, then we can say that the viruses are now different strains from one another. So this is a way of creating new viral strains, and that is antigenic drift. So that's kind of boring. Antigenic shift is far more dramatic, and that's why the name is such, because instead of the antigens gradually drifting apart, which is what's happening here, they're going to suddenly shift apart. So in antigenic shift, what happens is a single cell needs to be concurrently infected with 2 different strains of influenza virus of the same species. So that's important. In antigenic shift, we need 2 strains of the same species. So you can't have a strain of influenza A virus shifting with a strain of influenza B virus. That causes too radical a change, and that will just break the virus. So it's 2 strains of the same species that can produce antigenic shift. So 2 strains of the same species. So, for instance, if we have a human, and that human gets infected with 2 different strains of influenza virus concurrently, then potentially that human can be a place where antigenic shift is going to occur. But in order for it to occur, you need to actually get a respiratory epithelial cell, columnar epithelial cell, infected with the 2 different strains at the same time. So let's draw this in here. So here is the columnar ciliated epithelial cell here. And let's draw the 2 different strains of the influenza virus here. So here is one of them in red there, and we'll have the other one in an orange here. So these are the 2 strains of the same species of the influenza virus. Now, if they both infect this cell at the same time, then both of their genomes will go into the nucleus of this cell, and then when they're producing new virus particles, both of them will be producing copies of their genome. So the red one will be producing copies of its genome, and the orange one will be producing copies of its genome, so all of their ribonucleoproteins. And then what can end up happening is you can end up getting new virus particles budding off that are now a mixture of the 2 that have components of both of their genomes. So let's say we're talking about influenza A virus. So these are 2 strains of influenza A virus that are both capable of infecting humans and are both coming into this ciliated columnar epithelial cell at the same time. Each of them has 8 ribonucleoproteins, 8 strands of RNA with all of the proteins that accompany it. And when they're in the nucleus, they'll be producing new ribonucleoproteins, new copies of their genome, and what can then happen is they can get mixed up in the budding process. So you can take, for instance, maybe the first 4 ribonucleoproteins of the red type and the final 4 ribonucleoproteins of the orange type, create a new virus particle with that genome inside it, and that is now a totally new strain of the influenza A virus. Created all of a sudden. It's got a mixture of the antigens of the red one and the antigens of the orange one now. And this is the concept of antigenic shift, because we've suddenly created a brand new strain of the influenza A virus here, suddenly. The antigens haven't gradually drifted apart. This new strain has been created all of a sudden. It's a sudden shift. Now, it's not believed to be particularly dangerous for humans when you get antigenic shift occurring between 2 human strains in a human. The example that I've given you isn't believed to be particularly dangerous, and let me explain why. Because these are both human strains, and many people will have had both of these strains before, and therefore will have secondary immune responses to both of them, let's say. And then when they've created this new one, it's just got a mixture of the antigens of this one and the antigens of this one. So if you've got secondary immune responses to both of these, then you'll also be able to initiate a good secondary immune response to this, because the antigens of this are just mixtures of these ones. So if you understand what I'm saying there, that is why this isn't believed to be particularly dangerous when it occurs in humans. The dangerous thing is when it occurs in pigs. So this is where that diagram that I drew long ago, but I can't be bothered to find on this page, so I will draw again, becomes important. So a little set diagram here. So if this represents the entire population of influenza A virus. And by the way, this antigenic shift is particularly important in influenza A virus compared to influenza B virus and influenza C virus. This is where pandemics come from in influenza A virus. Antigenic shift can occur in influenza B and influenza C, but it's this non-dangerous form of antigenic shift. It's generally where two human strains concurrently infect the same human ciliated columnar epithelial cell, and then this creates a new strain of influenza B virus, let's say, or influenza C virus, but this has just got a mixture of antigens from the first two human strains, so many people will have secondary immune responses to this strain, and therefore will now have a decent secondary immune response to this, because all of its antigens are just from these two. Okay, so antigenic shift does occur in influenza B virus and influenza C virus, does create new strains of the virus, but it's not that dangerous because often people will have a secondary immune response to that if they've got these secondary immune responses to these old strains. Whereas in influenza A virus, because there is such a massive diversity of influenza A viruses, you can actually get new strains being produced to which people have not got decent secondary immune responses. So let me explain this. So this drawing here, this box, represents the set of all influenza A virus strains. So you can imagine putting loads of dots in here to represent each of the influenza A virus strains. So let me mark on now the subset that infects humans. So this is the human subset. So the dots, or the strains in this subset of the box, are the strains which can infect humans. Now remember, humans are not a big victim to influenza A virus compared to birds. Birds are utterly a huge victim to influenza A virus. There are absolutely a ton of strains of influenza A viruses which can infect birds. Far more strains of influenza A virus that can infect birds compared to the number that can infect humans, and these are called the avian strains of influenza A virus. Also recall the fact from earlier that most of the human strains are of the serotype hemagglutinin between 1 and 3 and neuraminidase between 1 and 2. Whereas the avian strains, you can have hemagglutinin between 1 and 16 and neuraminidase between 1 and 8. So there are a huge number of different avian influenza A virus strains, and they are markedly different, very different hemagglutinin and neuraminidase enzymes. Okay, but the overlap between the avian strains and the human strains is very, very tiny. Now, unfortunately, we have to introduce pigs onto this diagram, and the big problem with pigs is this. So here in pink, what I am trying to show with this is that pigs can get infected by a whole bunch of human strains and also a whole bunch of avian strains that cannot infect humans. So this pink box is representing the strains that can infect pigs. So look here, there are loads of strains that can infect humans and pigs. So lots of pigs can get flu that are human strains of flu. There's also this massive overlap here between the pig strains and the avian strains, so loads of the bird strains pigs can also get, and they can get a huge number of strains of bird flu that humans cannot get, this great big lump of this box here. This is what makes pigs dangerous, because now pigs can get infected by, let's say, this pink one here, and then this green one here. So let's say we have a pig, and I'll just draw a little pig here. This is not going to be a great picture, but here is the pig here. The pig, let's say this pig is going to be very unlucky. It's going to catch two strains of influenza A virus at once. It's going to get this strain here in green, which is a human strain that can infect pigs, and it's going to get this strain here in pink, which is a avian strain, which can infect pigs. And it gets both of them at once, and in its respiratory tract, it might be the case that a ciliated columnar epithelial cell gets infected with both the green strain and the purple strain at once, and then antigenic shift can occur just as described here, and you can create a brand new strain that is a mixture of green and purple, and it might just be the case that this new strain that you've created, and I'll give this evil red colour, so let's say the new strain that we've created inside this pig, strain red here, very evil strain, maybe this is a new one that can now infect humans, a brand new strain that can infect humans. Now, this strain has got a whole bunch of antigens from this avian strain here. I mean, some of its antigens will be from the human strain, but some of its antigens will be from the avian strain. And those antigens might be completely different. For instance, the hemagglutinin and neuraminidase might be completely different from the hemagglutinin and neuraminidase proteins that any human strain would ever have, and therefore the immune system hasn't got a secondary immune response to this and can't cope with it. So this way, inside pigs, we can believe is where new strains that can infect humans and to which no one in the human population is going to be immune to can be created. So, in pigs, we believe new strains of influenza A virus that can infect humans to which no one on the population is immune to can be created, and these are the ones that can then cause a pandemic. That's what you need in order to cause a pandemic. If you're an influenza A virus strain and you're looking for prestige, you want to cause a pandemic, you need to be, you need it to be the case that no one in the human population is immune to you, so that you can infect humans and then they'll all get a horrible disease, then they can spread it onto the next humans and spread it onto the next humans, and it can sweep across the population. The way that the influenza A virus spread would be stopped is if it suddenly reached a bunch of people who were immune to it, because they then wouldn't get a very severe disease and they wouldn't produce many virus particles, and therefore they wouldn't spread it on. In order to cause a pandemic, you need it to spread from person to person to person relentlessly and not get blocked anywhere. So you need the whole population not to be immune to it. And in order to create a virus that no one is going to be immune to, it needs to have radically different antigens now, so that you're not going to have a very strong secondary immune response. And I will just make a comment about this. Of course, this will have some antigens from the original human one, and you might say, well, won't you have a secondary immune response to that? Yes, you may well do, but that's not going to be very strong on its own. In order to get a strong secondary immune response, you need to have loads of them going off against loads of the different antigens of the influenza virus, all concurrently against loads of the different peptide fragments of the antigens of the influenza virus, if we're talking about T cells, or against loads of the different epitopes of the antigens of the influenza virus, if we're talking about B cells. So it's, you can't just have one B cell response against one epitope of one antigen of the influenza virus. You need to have loads of them concurrently in order to get a proper secondary immune response, in order to get a proper immune response full stop. So do understand that point, that we do need concurrent immune responses really to get it strong. Okay, so that's why how we believe brand new strains of influenza A virus can be created that have the potential to then cause a pandemic across the world. So the final question that I want to answer is, why do the influenza B virus and the influenza C virus then not cause pandemics in the way that influenza A virus can? And the reason for that is that you don't get the sudden production of a brand new human strain that is completely different from other human strains, like we've just seen you can get in the influenza A viruses population. In influenza B virus and influenza C virus, remember, there is much less variety of strains. There are far less influenza B virus strains and influenza C virus strains. The variety is much lower. Remember, for influenza B virus, the hemagglutinin and neuraminidase, they were all of one serotype. There was only one hemagglutinin serotype and one neuraminidase serotype. There's much less variety in the structure of those. Again, for influenza C virus, there was only one serotype of the hemagglutinin esterase fusion protein. So IBV and ICV have much less variety. So I'll just put this here. IBV and ICV have much less variety. And therefore, you can't produce brand new strains that no one has a good immune response to in the IBV and ICV population. And without that, you're not going to get a pandemic. So, of course, you can use Antigenic Shift and you can use Antigenic Drift in the IBV and the ICV population to gradually create new strains that will then be able to infect some people of the population and will cause full-on flu because they don't have a secondary immune response to it now. But then, without having the entire population susceptible, you don't get the spread that is a pandemic. It will just spread among the susceptible people who don't have a strong enough secondary immune response to bust it out quickly. But the other people who do have a good secondary immune response against these new strains, because of the fact that there's not as big a variety of these viruses, you can't produce a radically new one that no one is going to have a good secondary immune response to. So there will be people in the population who do have a good secondary immune response, and therefore it won't spread across the entire population in the same way. So I hope that is clear that why you get these brand new strains in the influenza A virus population that can suddenly cause a pandemic where it spreads across the entire world. And with IBV and ICV, even though you do gradually get new strains being produced through antigenic drift and antigenic shift, these new strains, they're not going to suddenly have no one in the population being immune to them. Some people will be immune to them, and therefore they can't spread in the same way as a pandemic strain would be. Okay, so that is the concept of antigenic drift and antigenic shift. So the next thing that I want to talk about in this video is vaccination against influenza virus. So let's now talk about the vaccinations. So at present, we vaccinate people who are at risk of getting flu, and who are at risk of getting flu and potentially not getting better from flu. So we vaccinate elderly people, we vaccinate people who have asthma, who if they get flu can get horrendous asthmatic attacks. We vaccinate people certainly with HIV or AIDS. We vaccinate people who are immunosuppressed and taking chemotherapy. So we vaccinate people who are at risk of getting flu and who are not going to get better if they do get flu. Now, how does the vaccination work then? Well, the vaccination is what we call a subunit vaccination. And this means it's going to contain proteins of influenza virus strains rather than the entire virus. And the proteins that it contains, you can probably predict which ones it's going to contain. It's going to contain the hemagglutinin protein and the neuraminidase protein. And now the next question you should ask is, okay, which hemagglutinin protein and which neuraminidase protein? Because we know that in the influenza A virus there are loads of different types of hemagglutinin protein and loads of different neuraminidase protein which are significantly different enough not to have overlapping antibody responses. Okay, we also know that in the influenza B virus, they've got their own hemagglutinin molecule and neuraminidase molecule, and an influenza C doesn't even have hemagglutinin or neuraminidase at all. So the first thing to say is we do not vaccinate currently against influenza C virus. We vaccinate against influenza A and influenza B. Now, in the vaccination, it's updated every single year, and we try and put in hemagglutinin and neuraminidase from strains that we believe are going to cause problems this winter. So what they do is they get hemagglutinin and neuraminidase from overall 3 strains of influenza virus. 2 of them are influenza A viruses. Now, the first one then is from an influenza A virus of the serotype H1N1. The second influenza A virus strain that we use is from the serotype H3N2, and then we pick a strain of influenza B virus, which of course won't have a serotype. Okay, so we'll have a serotype, but we don't give it a H and N number. Okay, so when we're creating this vaccine, and we do this every year, drug companies do this every year, and because it contains hemagglutinin and neuraminidase molecules from 3 different strains of influenza virus, we refer to it as a trivalent vaccine. So what they do is they firstly look at the influenza A virus population, and they start by looking at the influenza A virus strains of the serotype H1N1, and they try to find the strain that they think is probably going to cause the biggest problem this winter. I don't quite know how they predict that, but they have ways of trying to work out which strain they think is going to cause the biggest problem within that serotype. They take that specific strain and they produce the hemagglutinin and neuraminidase molecules of that specific strain, and they put them in this vaccine. So we're going to have hemagglutinin and neuraminidase molecules from a strain of influenza A virus of the serotype H1N1, specifically the strain that they think is going to cause the biggest problem this winter. Then they go to the H3N2 serotype of influenza A virus, and again they try to predict which strain of H3N2 serotype do we think is going to cause the biggest problem this winter. And they find that one, they agree on which strain they are going to use, and then they produce its hemagglutinin molecules and its neuraminidase molecules, which of course will be different from the hemagglutinin and neuraminidase molecules of the first strain that we're going to use, and they put those into the vaccine as well. So we've got 4 proteins so far. Then they go to influenza B virus, and they say, which strain of influenza B virus do we think is going to cause the most problem, i.e. make the most people ill this winter? And again, they take its hemagglutinin and its neuraminidase molecules, they produce them, and they put them in this vaccine. So the vaccine is going to contain 3 hemagglutinin molecules and 3 neuraminidase molecules. As well as that, it will contain something called complete Freund's adjuvant, CFA. This is something that all vaccines contain. So this is complete Freund's adjuvant. So this is a mixture of proteins from horrible pathogens, such as TB pathogen and other pathogens. And the whole reason that we put this mixture of proteins and molecules from horrible pathogens in vaccines is to trigger an inflammatory response. You see, if you just injected in these proteins into the body, you might not actually get an inflammatory response at the area of the vaccine. And if you don't get an inflammatory response, you're not going to get an adaptive immune response, because remember, the cells of the inflammatory response, the dendritic cells and the macrophages, they are the things that initiate the adaptive immune response. They initiate T-cell activation, and without T-cell activation, you do not get an adaptive immune response. Hence, you need dendritic cells and macrophages at the site where the vaccine is delivered, which is usually into a muscle or subcutaneously. And for that reason, we put in complete Freund's adjuvant, which is loads of molecules from horrible pathogens such as TB that will cause a massive inflammatory response at the site where the vaccine is given. Now, this is not dangerous. The live pathogens are not in there. It's just proteins and other molecules from the pathogens, but the live pathogens aren't there. So this can't cause you disease, but it will cause you a massive inflammatory response at the site where the vaccine is given. And that means that we're going to then get adaptive immune responses initiated against the 6 proteins that are coming in with the complete Freund's adjuvant. Okay, so how will this protect you then? We are going to give you a vaccine with these 6 proteins in of these free strains of influenza viruses with something to actually cause an adaptive immune response. Now, the type of adaptive immune response that you are going to get is only going to be the B-cell response. You will not get the cytotoxic T lymphocyte response in response to these, because these are not going to be inside cells. These are extracellular molecules that will be phagocytosed by dendritic cells and macrophages, and therefore peptide fragments will be loaded on MHC class 2, not MHC class 1. So you're only going to get the B-cell response, but the B-cell response is incredibly powerful. It will result in antibodies being produced of the IgG and IgA class against these 6 molecules, against the hemagglutinin and neuraminidase molecules of these 3 strains of influenza virus. The IgA molecules are then, of course, going to end up in the mucus. So we're now going to have in our mucus antibody molecules, IgA molecules, against hemagglutinin of the H1 serotype of influenza A virus, against the H3 serotype of hemagglutinin of the influenza A virus, and also of the influenza B virus. And that, for 3 months or a few more, for a few months afterwards, is going to be in a high concentration in the mucus, and therefore is going to be able to neutralize a huge number of influenza strains that you might well breathe in and which might cause flu if we hadn't had the vaccine. So it's going to be able to neutralize a huge number of influenza A virus strains of the hemagglutinin 1 serotype and of the hemagglutinin 3 serotype, so it's going to protect you against a lot of strains of influenza A virus, the major strains that can infect humans. For some reason we don't do H2, I don't know why, but this is the way it's done at the moment. And it's also going to protect you against a huge number of strains of influenza B virus. Remember, in those first few months when you've actually got the antibodies in the mucus protecting you, it will be against not just the strain that you've been vaccinated against, the specific strain, but also against strains that are very similar to these ones. Okay, after those few months, then of course the way it will be protecting you is through the secondary adaptive immune response that you will get if you do get exposed to one of these strains of influenza virus or strains that are similar to it. And that's the way this vaccination works. And every year they update it so that the strains that they are using are believed to be the ones that are going to cause a problem this winter. And of course, if we use the actual strain that we think people are going to get infected with, that will give us the strongest secondary immune response. I've talked about how if you vaccinate someone against a specific strain of H1N1, it will protect you against more than just that strain, but other strains that are very similar to it. But of course, the actual strain is the one that the secondary immune response will be best working against. So it makes sense, therefore, to try and boost up the secondary immune response ready for the actual strains that we do believe are going to cause a problem this winter. So that's the way that the trivalent subunit vaccine for influenza actually works at the moment, at the time of making this video. In future, who knows how many different strains we'll be vaccinating against in the influenza vaccine, but this is the way it currently works. Okay, so we'll have a break here. In the next video, which I think will be the final video, we will discuss antiviral medications, the neuraminidase inhibitors and the adamantane medications, as well as symptomatic treatments. So we'll talk about paracetamol, ibuprofen and aspirin, and then we will end this video.